Well, it is my great pleasure to introduce you to Dr. James Tour. I think he would be better suited to give his credentials than I would be. I definitely would fumble over many words. So, welcome, Dr. Tour.
Thank you so much. Can you hear me okay?
You're coming through loud and clear.
Okay, good. Yeah, let me. Okay, so yeah, I'll probably address some of the questions that just arose. Um, this is probably one of the very few times I'm giving a talk where I am in a friendly environment. So it's a bit unusual for me. I usually come locked and loaded. So if I'm a little too intense, it's just because of seasoning for me. Um, okay, but we'll begin. We'll talk about this whole thing of abiogenesis, and scientists are still clueless on life's origin. And I'll be appealing to researchers in the field. Um, so with that, I'll begin here. And so here's a list of different researchers in the field: Professors Cronin, Benner, Sostek, Joyce, Sutherland, Lane, Lancet, Deemer, Reichert, Lipschitz, Hudd, Krishnamurthy, Devaraj, Pauner, Rout, Morey, and Walker. And I would just say, let us reason together. I actually think that these folks, other than Sarah Walker, who is not an organic chemist, she's a physicist, so I don't think that she really sees these things. But everybody else here is an organic chemist. And I think they actually see what I see. I don't think I'm alone in this. I just don't think that they're coming clean with their thoughts. I don't think that they're expressing their thoughts honestly. That's just my view. And if they disagree, I know they're not watching now, but if we get this recording, we can post it in places where they see it. And I have the utmost respect for them as chemists, and I think they actually see what I see. In fact, when I've asked some of them, when I've challenged them, I said, you see what I see. They don't contest, they just don't answer. And remember, no answer is an answer in itself. So, definitions. Abiogenesis is the origin of life from non-living matter. To construct any convincing theory of abiogenesis, we must take into account the conditions of the Earth about 4 billion years ago. It's not my definition, that's the dictionary definition. So we're going to think about what an early Earth must have been like before there was life on the Earth. And we have to make life from non-living matter. Prebiotically relevant means that we're restricted to materials and procedures and conditions that might have been available on an early Earth. So we had no chemical manufacturers, we had no human-made synthetic methods. We had only very simple reactions that might have occurred naturally. We certainly had meteorites that could have brought a lot of material to Earth, but those meteorites almost always bring vast mixtures. We've never had a meteorite just bring a singular compound. Most of what comes to Earth is already burned up, but in a few cases when it's been buried deep within the meteorite, you can have compounds, but they're usually in very small amounts, and they're usually in mixtures of compounds that could never be used, because if you have a hydroxyl, an alcohol functionality on one molecule, and then on a different molecule you have hydroxyl functionality, both of those are going to compete. If they're both secondary alcohols, they're both going to compete for the same types of reactions, so you have competition of reactions. Nobody was present at life's origin, so we'll never really know how life originated as scientists. But that's not what we're seeking to answer. Actually, what we're seeking is an experimentally verifiable hypothesis as to how life might have originated. That's what we're seeking to answer, actually. So what are the textbook characteristics of life? Its responsiveness to the environment, its growth and change, ability to reproduce, have a metabolism and breathe, maintain homeostasis and being made of cells, and passing traits onto offspring. Homeostasis is a steady internal physical and chemical condition. This is, again, not my definition. This is the Academy's definition of life. Now, people are trying to now redefine life to make it into, in a sense, their own image, into their own target, so they can say that they've hit it. This is the target that we've got to deal with. It has to have cells. It has to pass on traits to offspring. It has to have this amazing internal state that biological systems have, which is to be extremely low entropy while being very high energy. So a high energy system and low entropy or low disorder. Things have to be in a very ordered type of condition. It's a very interesting combination of things. People will say, oh, well, tornadoes have those characteristics. They do in a sense, but not nearly with the control of a cellular system. They're unusual, even in nature, unless you go to a biological system, a cellular system. I'm not going to speak of God of the gaps, none of that. So any accusations that some may levy against me that I'm using a God of the gaps argument is a lie. I'm not using a God of the gaps argument. In fact, people who often contest with me use a time of the gaps argument. They have no explanation, and so they will call upon large amounts of time just grabbing that out of nowhere and suggesting that time explains lots of things, whereas actually time is the enemy. Time is the enemy because time results in decomposition, especially on an early Earth where you would have so much UV radiation. But even without that, there was a question that came in just in the last session about water. Water is extremely degradative to organic compounds, condensation polymers. Condensation polymers are the way that all of the biological polymers are made, which means that you release water on their synthesis. That means that they don't tend to go in water without the help of enzymes, so there were no enzymes around. So they don't tend to go. In fact, the free energy is positive, means they go the other way. So water prevents the polymerization of all of these species. And water causes the degradation of these species. And we're going to talk in a little bit what happens when you just happen to have one molecule. So these are the molecules that are the language of living systems. You have to have four basic building blocks here. You have to have your monomeric sugars. Wherever there's a red line there, the red arrow, those are stereogenic centers. You have to have an amino acids. 19 of the 20 canonical amino acids have a stereogenic center. You have to have nucleotides. Those bear 4 stereogenic centers, both in DNA and in RNA. And you have to have, in RNA, in DNA, you would have 1 less stereocenter. And then you have to have glycerol, but you have to be able to do what's called enantiotopic distinguishing. You have to have you have to distinguish between these 2 hydroxyl groups, because the reaction that occurs here at one of this site or this site is going to then define whether this center is going to become enantiotopic, an enantiomer, which means that these 2 centers are enantiotopic. There's no stereochemistry there now, but as soon as one functionalizes, boom, that becomes a stereogenic center. That's got to be right. Now, these have to polymerize. The polymerization is the thing that always, always, always stops the materialist directly in their tracks. They will argue that monomeric sugars can come from the foremost reaction. They are wrong. They can come from a foremost reaction, but never in impurities that are useful. You get billions of polyols, billions of oligomeric alcohols in a foremost reaction within minutes. So those would be totally unusable. But they'll say, oh, well, these may come on meteorites. I'd like to see these come on meteorites with any purity. Amino acids, these could come, they will argue, from the Miller-Urey reaction. Yeah, they come. but with many, many other compounds, many carboxylic acids that would compete. So it's useless. Mixtures are useless. Biology is amazing because you can take a massive amount of molecules, and enzymes which are handed can go in and pick out a particular molecule. You can't do that in chemistry. You can take a handful of supplements for breakfast, and your body knows how to segregate each of those. You can't do chemistry like that. Never works that way. You take a handful of many things, mix it together, you can't do chemistry because they all compete with each other. You got to solve the sugar problem before you can make a nucleotide. And you got to be able to do this in anti-atomic chemistry. But this polymerization, how do you do this? Free energy on all of these is positive. How do you do this? This is actually going to be DNA here. If we polymerized actually this one, it would go to RNA. And then lipids. RNA is quite unstable in water. It doesn't last very long at all. We'll look at some of the timing on that. And then how do you do this distinguishing? So the polymerization stops them in their tracks. They have no answer to this. Hot-dry cycles doesn't do it because what happens is you get racemization faster than you get oligomerization. So you'll lose all your stereocenters, even if you could distinguish these. Nobody knows how to insert just one stereocenter. And even if you had them separated, faster than you would polymerize these, you would start racemizing the centers on the oligomer that you started making. And there's no way to present the side chain reaction. You want a reaction here, you want a reaction here. You don't want it here. No way to know how to do this. Dead. It's over. You can't make polypeptides. Abiogenesis, you can't make polypeptides, so you can't make any enzymes. It's toast. Polysaccharides, there's too many combinations here, which we'll see. Okay, molecules don't care about life. They've never been known to move toward life, ever, unless there's an organism, unless there's like a person or a cell acting upon those molecules. But chemistry by itself, molecules, they don't know to move toward life. They don't say, hey, let's try to make a cell. They have no brain. They don't move toward life. They actually move the other way because of entropy. They move the other way. They don't move toward a very low entropy system. Has the public been misled on origin of life claims? Absolutely. The question was asked, under simulations of early Earth's atmosphere, scientists have mixed molecules together in laboratories to produce complex life forms such as frogs. 1/3 of the general public believes that is true. How about simpler things like a bacterium? Under simulation of early Earth's atmosphere, scientists have mixed molecules together in laboratories to produce simple single-cell life forms such as bacteria. 2/3 of the general public believes that is true. I'll tell you the answer to both of those is really false. So who is that general public? Well, the average age was 38. They're half male, half female. 80% of them hold college degrees at some level. That's how badly the general public is confused. Why might they be so confused on this? Why might they be so wrong? Who, perchance, may have confused them? Well, there's been real mixed messaging, and the source of the confusion the genesis of the confusion is the origin of life researchers themselves. It is not the press. The press ramps it up, but the genesis, the origin point of the confusion comes with the origin of life researchers. Jack Szostak, Nobel Prize winner, was at Harvard, now at the University of Chicago. He said, he said on June 2nd, 2014, he said, "You heard it here first," announced World Science Festival moderator and astrophysicist Mario Livio, as Harvard biologist and Nobel laureate Jack Szostak told a Search for Life gathering of 50 on Saturday afternoon in New York that he expected to make life in the lab in 3 to 5 years, and more likely within 3 years. That's what Jack Szostak said. That was in 2014, and let me just reveal here, he missed his deadline. Even if we give him the 5 years, he missed it. He hasn't made life yet. Harvard astronomer and Origin of Life Initiative director Dimitar Seselov, also a panelist, told the audience that years ago, Origin of Life was a neglected question because most people thought it was too hard. But outside the meeting room, he confirmed that we are indeed now very close to life. Seselov thinks it will take 5 years, not 3. Seselov missed the deadline. Jack Szostak, to a professional gathering in 2021, said this, and I give you the URL link. He said he's not even yet made the RNA in a prebiotically relevant manner. It hydrolyzes too rapidly. That's the summary, but what he actually said, his words are, and basically that's because RNA, we know we had to get RNA. And RNA is delicate. It also has a phosphodiester backbone. It is subject to degradation by intramolecular attack of the 2' hydroxyl on the phosphate. So you get cleavage of the backbone, which is catalyzed by metal ions. So we don't have millions of years. Everything actually has to happen pretty quickly. The synthetic processes have to be faster than the degradative ones. That's what he said. He's told this to a professional audience in 2021. Why didn't he tell the audience, the lay audience in 2014, that he hadn't yet even made the RNA? And RNA is far from having life. That's the mixed messaging. It's Dr. Stephen Benner, former Harvard professor and now director of the Foundation for Applied Molecular Evolution, said to a YouTuber in 2021, I suppose many of the big paradoxes in Origin of Life have been solved. Oh, really? I would contest with this. I would say that none of them have been solved. Not one. Name for me one of the paradoxes in Origin of Life that's been solved. Professor Benner, at a professional meeting, said this in 2019. He said, chemistry is actually hard to get to work. The molecules precipitate, the molecules hydrolyze. The molecules decompose, and so it's very much a constraint that you have to deal with. It's one goddamn problem after another. That's the truth. That's the truth. The molecules hydrolyze. Water is a very hard solvent to do these polymerizations in because the free energy in water is positive. Unless you have activation, which occurs like with an enzyme, and then unless you have blocking of all the other competing moieties, you don't get it. You don't get what you want. Not going to happen. It's a huge problem here. And so these have not been solved, but he tells the YouTuber most of the problems have been solved. Is mixed messaging. Professor Cronin in a TED Talk in 2011 said, what I'm going to try to do in the next 15 minutes or so is tell you about an idea of how we're going to make matter come alive. Wow, that's interesting talk. He was asked by the moderator, Chris Anderson, at the end, just a quick question on timeline. You believe you're going to be successful in this project? When? Lee Cronin, hopefully within the next 2 years. That was in 2011. He missed his deadline. None of these guys are saying this anymore, but that's the source of the problem. It's them. Sadly, comments like this from researchers are misleading to the public. That's where the problem is. That's where all this public perception has gone wrong. All 3 of these researchers have more recently backed off using these inflated statements because if they use it again, I'm going to project it all over the place. What's the real state of Origin of Life research? Well, nobody has shown a method to make the enantiomer versions of carbohydrates, amino acids, nucleotides or lipids in a prebiotically relevant manner. Nobody's ever made these. Nobody has ever made these in a prebiotically relevant manner. Miller-Urey didn't. Those are totally unusable. Remember, even people say he may have made up to 10 or 12 of the canonical amino acids. They were all racemic, but let's just say somehow he could use them racemic. There's hundreds of other compounds that he had in there that would compete, that would make all of those useless. You can't just take a mixture of amino acids and do chemistry with those things. You just can't, unless you have enzymes to select them out. Carbohydrates, nobody's ever had them. Nucleotides, don't have them. Lipids, you don't have them. And remember, diacetyl lipids, you don't have it. Once you functionalized one end of that glycerol, you functionalized one end, that now becomes a stereocenter. Nobody's ever controlled that stereochemistry. Big problems. So nobody's ever done that. Nobody's ever shown that life could form with lower enantiomeric excess mixtures, thereby mitigating the need for chiral-induced spin selectivity. Chiral-induced spin selectivity was discovered about 25 years ago. And what it shows is that we were taught that enzymes worked because of like a hand and a glove. A right-handed hand goes in a right-handed glove. That it was all these stereoelectronic arguments of a fit, of a substrate within a cleft. That's what we were taught. That's only half of the story. There's another half. The other half is this, that when you have a chiral environment, the electrons, so everything is going to start, all reactions are going to start with electron transfers that you have from one molecule to another. But when you have these electron transfer interactions, you have these electrostatic potentials. And what happens is only certain electrons go and others don't, because electrons can be spin up or spin down. All chiral environments are spin valves. So only one electron spin is selected and not the other. What's the ramification of that? The ramification of that is that when the proper spin is coupled with the proper chiral environment, you don't have backscattering and the electron goes through much more easily. If you had the wrong electron going through, it would backscatter and you had to have much more heat release. And this is why we don't burn up. And this is why my computer chip is very hot, because it's not selecting the spin on the electrons, whereas I don't burn up. It's because of this chiral-induced spin selectivity. And it's this is why in nature you can have two hydroxyl groups go to form hydrogen peroxide, because they'll go through an anti-parallel singlet surface. Or they can go through a parallel triplet surface, and the two hydroxyl radicals can form oxygen and 2 moles of water, 2 moles of proton. So you see, the same substrate can go through two totally different pathways in a biological system with 99.99% selectivity in this because of the chiral environment. So it's not like nature could have optimized later and refined these to be chiral systems. You had to have chirality before nature ever began, before biological systems ever began. Nobody's ever shown that the mixtures found in meteorites or interstellar space could be useful for synthesis. Even John Sutherland, probably the top of all the origin of life synthetic chemists, he even says, I don't think meteorites make the right sort of mixture, but we need to have more constrained chemistry to actually make the right sort of mixtures. You can't just take all these random compounds, like even Miller-Urey. I mean, there's too many competing compounds. You get so many other amines in there that are not alpha amino acids in his mixture. You get so many carboxylic acids in there that are not part of amino acids, that are not part of an alpha amino, the type of amino acid that you're going to need. So it's not going to do this. It's not going to be this alpha amino acid form. So meteorites, that's why I say, when we have these discussions, I'll give you all the small molecules you want. I'll even give it to you in purified form, because they can't make the polymers anyway. So let them think that they have this. They don't have it. Nobody has solved the mass transfer problems in chemical transformations from small molecules to a cell. How many steps are you going to need to go from small molecules to a cell that shows responsiveness to the environment, growth and change, ability to reproduce, metabolism? This is going to be thousands and thousands of steps. Well, what happens if you had 1 gram of each of these molecules, a gram of carbohydrates, a gram of each of the amino acids? You're going to run out, because your reactions are not going to be high yielding. All right, so what if you had 1 kilogram of each of them? How much could you bring through? You run out of starting material, so you always have to bring up starting material from the rear when you're doing a synthesis. But when you run out of starting material, what do? You go back and you look at your optimized procedure in your notebook and you know how to go back through it. In nature, you've gone on for, say, 500 million years building this. You've run out of material and you're like, oh no, I got to go back and make some more because I ran out of material because I just used it all up on this last step, which failed. Can't go back. I mean, it just went to tar. Well, just go back and make some more. But nature says, how do I make more? I never kept a laboratory notebook here. I don't know how to go back. Too bad. Too bad. Start fishing around for another 500 million years. You can't do it. You could start with a ton of each of these small molecules. You're going to run out because these are not optimized reactions. These are just floating around type reactions. They don't know what optimization is. They don't know why to stop. They don't know what the product is. They don't know where they're going. So no one solved this mass transfer problem. This is a big problem when you do total synthesis in a laboratory. Nobody solved the amino acid polymerization problem with amino acids bearing active side chains. So even if you could have an alpha amino acid that is polymerizing from beginning to end, what happens with these functional ones? All the one in red are very reactive. You have hydroxyl groups here. You have a thiol group here. You have another phenolic group here. You have here's a carboxylic acid. How does it know the difference between this carboxylic acid and this carboxylic? It doesn't. All of these would cause cross-linking. Then you have somewhat reactive side chains in the dashed circles. All of those are going to cause cross-links. Once you get one cross-link, it's dead. The thing's going to crash out. It's dead. It's over. Game over. And the more of these you get with these active side chains, the more chance that these active side chains are going to be participating rather than those two tiny little ends that you wanted to participate from. Nobody's ever solved that problem. Nobody even addresses that problem. Why not? Because they can't. So I posed to them this question. Let's take making a polypeptide off the table. So this was a question that came up just before I started talking. And I posed this question, just make DK, just make aspartic acid coupled to lysine. In fact, I will even take aspartic acid coupled to lysine, so this carboxylic acid and this amine, or you could give me this carboxylic acid coupled with that amine. I'll take that. DK or KD, I'll take it. Could you even make the dipeptide in, say, 90% yield or higher? Because it's going to have to be good yield. Remember, if you don't have high yield here, the fidelity kills you. Well, how are you going to stop this carboxylic acid from competing with these two? How are you going to stop this amine from competing with that one and that one? You can't. You can't. How do you do it in a laboratory? You selectively do protection. You block this with a selective group. You block this with a selective group. You block this and you block this, and you only get reaction there. And then you have to deprotect. No way to do that without human beings being around to do it, or selective enzymes there to do it. They're lost. It's not even that they can't make a polypeptide. They can't even make a dipeptide with the needed stereo control. Plus, the free energy here is all positive. This doesn't go. It will revert back the other way. Water hydrolyzes this much faster than this goes forward in water. So the free energy is positive on this. So they're dead. It's game over. Nobody solved the nucleotide polymerization problem. How do you couple this so that you have the 3' hydroxyl coupled to the phosphate on the 5' position of the next one? 3' to 5', 3', so forth to 5'. This is a 2' coupled to a 5'. Why? So now you've got this 2' alcohol coupling. It's game over. Nobody's been able to do this. Every polymerization that is known to do this on the surface of crystals, on rock interfaces, outside of an enzymatic environment, has had 30 to 70% of the 2' 5' linkage. Game over. It's toast. It's over. Plus, how do you prevent this 2' hydroxyl from doing another polymer off of this end? That's the branching problem. Nobody has solved that outside of a biological environment. Never been solved. It's game over for them. If I gave you a nucleotide and you can activate, I'll even give you the triphosphate or even phosphorimidazole. That way you can have it even activated. I say, could you polymerize this to a nucleotide with less than 2% of the 2' 5', or less than 2% of the branching? Because 2% is crazy high. Remember, 2%, you might find a run of 50 of these being undisturbed. So 2% could you do that? Nobody can do this. I've posed this question to the vast majority of the people that I've called out in the beginning. I've posed this to them. I said, if you can do this, if you can just show me how this can be done outside of biological environment, I'll stop talking about Origin of Life. I won't do the Creation Summit anymore. I'll take everything off my website. None of them could do it. Why didn't they just step forward? Because they can't. Game over for them. Nobody solved the carbohydrate polymerization problem. If you have 10 A units, if you have 6 A units, how can you hook them together? A. You think that you just want to make this in a run. How do you do this? Well, if you just had 6 glucose molecules, because all of these hydroxyls are potentially active in hooking up, you can hook up 6 glucose molecules in 1 trillion different ways. More than 1 trillion different ways you can hook up these 6 glucose molecules, because you can have open form, closed form. You can have 5-membered ring, you can have 6-membered ring, and you can have all of these coupling, and then you can have the alpha and the beta form at the anomeric center. So that's what you're up against, over 1 trillion combinations just to make a hexamer. If every ailment in a human body, every ailment in a cell can harken back to an accompanying carbohydrate coupling problem. You get the couplings wrong on this, it's game over. Nobody knows how to couple the carbohydrates. That's what I'm telling you. The polymerization just doesn't go. Plus in water, it doesn't go. The free energy is going to be positive. Dead. It's over. Game over for them. All right. Let's not even think about a polymer. Could you just take glucose and make a disaccharide? So I challenged all these chemists. I said, could you just make a disaccharide? Just make this disaccharide in 90% yield or greater for me in a prebiotically relevant manner. If you do that, I challenged all of these science. I'll take down everything. I'll never talk publicly about Origin of Life again. Guess what? They couldn't do it. They couldn't do it. So forget about polysaccharides. You can't even make a disaccharide. It's over. Game over for them. This is what nature has to go through to make glucose. Just that simple molecule glucose, just this simple molecule glucose, this is what nature goes through. You've got 11 different enzymes, 4 of them unique to glucose synthesis, plus 4 activators. So you've got all of this. What's the chances of this thing forming randomly? 10 to the 6,368, because it's 20 to the 4,700 amino acids. And so you take 4,700 log 20, log of 20 is 1.3, give you 638. So it's 10 to the 638, 10 to the 6,368. So just for making this one. So this is not going to form. Remember, if you're over 10 to the 40th, there's not enough time in the universe. So this is a lot bigger than 10 to the 40th. And then you have to have made this one and this one and this one. So it's like 10 to the 65,000, the chances of making all of these. And remember, they're going to have to be around the same place at the same time, because any one of these intermediates is going to fall apart. So they've got to go quickly from one to the other, to the other, to the other. And these are the steps it has to go through. You can't make glucose. And then when you make glucose, how does nature polymerize the glucose together since it's so hard? Well, it uses phosphorylase, uses this enzyme. That's 842 amino acids. So you got 10 to the 1094 possible combinations of that many amino acids. Not going to form. There's not enough universes to do this. It's not going to happen. So this is the problem. This is the problem. game over for these guys. Now, the question was, are these stable in water? The answer is no, because let's just say you got an RNA. Let's just say an RNA formed that was, say, a 600 mer, so it could code for a 300 mer protein, which would be an enzyme that would do something. If you don't have a mole of these, remember, you don't have a mole, you only have one of them. One happened to form. All right, so if it formed with the right sequence to do this type of thing, how long would it last? So you can't use half-life. Half-life is when you have a lot of entities. You have a mole of entities, what's the half-life? So after that half-life period, you have half of what you had to start with. Well, a 600 mer, you now have to convert a half-life to a probability. And when you do that, and I've just written a paper on that, which I've submitted just to show the math here. It's really very simple. And then with my colleagues, we've shown it a separate way using a different method. But if you have a 600 mer of identical RNAs with a half-life of, say, 100 days, that's very generous for RNA in water, that it would have a half-life for 100 days. If you had, say, a mole of them, well, so that's 2,400 hours at room temperature in water. That's without any magnesium ions around, no divalent ions around. For a single RNA molecule, its lifetime is 2,400 hours divided by 600 is 4 hours. That one molecule would last 4 hours. Your time element is gone. You don't have time. As soon as these molecules form, now you have to get that RNA, you have to form that RNA. So you have to have just those right nucleotides around, no saccharides around, no amino acids, or else these are condensation polymerization. So you have to have very high purity monomers. So now they're all polymerized. Now all of a sudden you have to now bring to it all the 20 canonical amino acids that it's going to have to code for with the enzymes to help make that thing, and have all of that done in 4 hours. You can't even do it that fast in the laboratory. This stuff is decomposing. If you had a 200 mer polypeptide in water, its lifetime is about 13 days. If you had the one molecule, say one enzyme happened to form, and then life took off, which is a bunch of nonsense because life is much more than just one enzyme. Let's just say that one enzyme has to now find the substrate that it's going to act on in 13 days, or its history. Because there's only one molecule. There's not a mole of molecules. Nobody's explained how the requisite protein folding could have occurred. That's the Leventhal paradox 1.0. This is written in this article right here. Even a single protein could not arrive at its native structure in biological real time by random search because conformational space is far too vast. 10 to the 95 possible conformations for a chain of just 100 residues, so that even a small protein that initiated folding by random search at the time of the Big Bang would still be thrashing about today. Remember, if you're more than 10 to the 40th, there's not enough time in the universe. Nobody has explained how the requisite interactome could be prepared. This is the non-covalent interactions. So just molecules non-covalently are interacting. If you just think of the protein-protein interactions in a simple cell like yeast, a simple yeast cell, just the protein, we're not talking protein DNA, protein RNA, just talking protein-protein interactions. An interactome is the whole set of non-covalent molecular interactions in a particular cell. If one merely considers protein-protein interactome combinations in a single yeast cell, the estimate is 10 to the 79 billion. Remember, 10 to the 90th is the number of elemental particles in the universe. And when I say elemental particles, do I mean elements, or do I mean the number of protons, electrons, and neutrons in those elements? It doesn't matter. It doesn't matter. Because if you have an element, say it has 100 electrons, protons, and neutrons, we're only talking about 2 orders of magnitude difference. This is 79 billion orders of magnitude. So it doesn't matter what I'm talking about here. Nothing works. Nothing works. And so this is not my calculation. This is from these biophysicists. This is just crazy. This is what you have to order. So how does a cell do this? It takes this information and puts it to both sides of the cell, and then it necks down and you got your two cells. But the information came from the mother cell. I mean, it's just amazing. Nobody solved the code problem for ordering the nucleotides or saccharides or proteins. Every polymer in nature has a code, not just the polynucleotides. In fact, there's more information that can be stored in the polysaccharides than in the polynucleotides. More information in the sugars than there is in the DNA. Because remember, you have 10 to the 12, you have a trillion combinations of just 6 glucoses. So there's a lot more information can be stored in that. Every polymer is coded in nature. Nobody knows where the initial code came from. So nobody's ever made any of the higher order structures. You need lots of structure to create a cell. It's not just making RNA. This is the minimal gene set these biophysicists figured out in the simplest of cells. Computationally, what is the simplest cell you could have? And this didn't include any of the amino acids. It has no ability to make any of the amino acids. You'd have to bring all of these in exogenously. So the simplest of cells. Remember, you have to have DNA replication. You have to have repair. You have to have restriction. You have to have modification. You have to have transcription machinery. You have to have amino acyl tRNA synthesis. You have to have tRNA maturation and modification, ribosomal proteins, ribosome functions, maturation. How many of these have been made? How many of these have been made in a prebiotic sense, meaning that you didn't start with nature's constructs and scientists made any of these? How many? Zero. None of these have been made. None. You're going to have to make all of these to make your cell. And these guys saying that they're going to have life in 3 to 5 years is an out and out lie. That's what it is. You have to make all of these. How many of these have been made by those researchers? None. Remember, they didn't. Sostek, the RNA specialist, has never even made any RNA. None of them have ever done this. And you got to have all of this. You have to be able to build all of this. How many of these things have been built in a modern laboratory with all of their techniques without using nature's constructs? So in other words, they built up their own saccharides using modern technique. You know how many of these have been made? None. It's a problem. All right. So you professors, come now. Let's reason together. Why aren't you agreeing with me that we don't know what we're doing here? All right. So you take whatever model you want. Take the RNA world hypothesis. Take the protein world hypothesis. Take metabolism first hypothesis. Lipid world hypothesis. Hydrothermal vent hypothesis. Amyloid hypothesis. Go ahead, you could choose the ones that you've disregarded now, iron, sulfur, clay hypothesis, whatever you want. Take it. Choose your favorite. Tell me how you're going to make life. Just walk me through the steps. Walk me through the steps. I'd love to see this. Walk me through the steps. I won't even interrupt you unless I don't understand something. Walk me through the steps. What do you need to make a simple cell? You haven't made any of this. But what I'll do is I'll give you this. I'll give you all the small molecules you want, all in separate bottles, all the 20 canonical amino acids in separate bottles, the 19 that have a stereocenter. I'll give it to you with complete enantiopurity. I'll give you all the monomeric sugars you want in separate bottles. I'll give you all the glycerol you want, and I'll give you all the nucleotides you want, all of them. Now just polymerize these for me. Oh, you can't do it? Of course you can't do it. Free energy is positive. You can't do it. You can't stop this cross-linking. You can't do it. Okay, what I will do then is I will give you all of these. I'll give you all the polymeric forms. And these questions have been posed to them. I'll give you all the polymeric forms, and I'll even give you in any arrangement you want. So you have the code. So you have all of the information. So I've really conceded a lot here. I've given you the informational code because I've given you all in the order that you want. I'll give you all of these. Could you now take these and just assemble for me a cell? My question is, assuming you had access to all parts in your laboratory, not on an early Earth, not in some hydrothermal vent, but assuming you had all of this in your lab, how would you make even a simple cell? Remember, it's got to be responsive to the environment, growth and change. How would you do it? Just throwing all these parts together is like taking all the parts to a car and just putting them in a stack and saying, here's your car. No, that's not a car. No, it doesn't work. You shake that up all you want. It's not going to form a car. Shake this up all you want. It's not going to form a cell. Nobody knows how to assemble these. So how on earth can they suggest that their stupid little experiments are going to make a cell? Crickets. That's all you hear is crickets from them. And then you get this guy, Lee Cronin and his colleague, Sarah Walker. And they come out with this assembly theory, which they say is going to quantify selection and evolution. And they go around how this is going to explain something on origin of life. I don't understand it. And then when I met with Lee Cronin at Harvard, I said, watch, he's not going to address anything on origin of life. And he didn't. I spoke at Arizona State University last spring. And I told my host, I said, I'd love to meet with Sarah Walker when I'm there. Because she's there. And because she's talked about me on the internet, she says that I pose things that are irrelevant, straw men. When did chemistry become a straw man? So I wanted to meet with her. And my host said, no, she can't meet with you. So I contacted her myself. I said, look, I'm going to be on your campus. Could I please meet with you? No response. So I said, how about, would you let me meet with one of your students or your postdocs so that I can understand from them how you see this, doing anything for the origin of life? She wouldn't let me meet with any of them. None of them. Silence. Crickets. Why is it? Why is nobody will engage me on this? It says it's a groundbreaking new theory of everything. It unites physics and evolution. That's what was written. Assembly theory, bridging physics and biology to decode evolution and complexity. And this is, a picture from his paper where he's got molecules being built. And I challenged him at Harvard. I said, how do you get this to assemble? These aren't just Velcro balls. I mean, how do you overcome? Then he gets up and he says, this wasn't intended to be molecules. In your Nature paper, you drew molecules. You drew amino acids going to polypeptides. How could you tell me now, when I have you confronted here at Harvard with the Harvard and the MIT faculty here, that you can say, how dare I suggest that you're suggesting that it's with amino acids coupling? Because you put it in your paper. And that he talks about this causal history. If you can look at a molecular structure, and if there's more than one of them, if there's 2 or 3 or 4 of them with a certain number of stereocenters, you know that has a causal history. What is causal history? You mean that the Discovery Institute would say you had intelligent design, and other people would say you had God. Just because you write causal history, do you think that takes you away now? And now you can get away with just suggesting this after you've pummeled these guys for all these years? No, I'm not going to let you do that. And this is just Velcro balls sticking together. This has nothing to do with chemistry. There's nothing there. There's no chemistry. There's nothing there. I can look, if you show me that there's 3 molecules of, say, morphine with all these chemicals, that had a causal history. Because that could not have arisen just in an explosion just by itself. Something happened. I don't need your little silly equations to tell me that. I can look at a molecule as an organic. I can look at molecules. You tell me you have three or 4 of those. I can tell you that had a causal history, if you want to use those words. That didn't just spontaneously assemble like that. So Professor Cronin now argues that my 60-day challenge, when I gave him 60 days to answer these questions that I had said, just show me one polymer, show me the dipeptide, show me the disaccharide. He said, he said, quote, they're actually connected together, and you should start. I'm sorry, let me start reading at the top. He says, a simple cell started somehow, and evolution selected for information and the subsequent reactions, or it all started at the same time. So in other words, he says the cell was there somehow, and then evolution started to select for this. He says they're actually connected together, and you should start at the base, meaning the cell, and this is why James, referring to me, is doing it the wrong way around. So I'm proposing the wrong way around, molecules having to build up into higher order structures, and then you get a cell. No, he's suggesting somehow there's a simple cell, and then refinement of the information, and then the selective chemistries to the three classes of polymers that derived the polysaccharides, RNA, and polypeptides are all happened at the same time. That's what he's suggesting. You go to this link. You go to this link and you will see him suggest that. He says, James had it the other way around. You actually have the cell and the YouTuber just sitting there. Oh yeah, okay. I mean, how does that work, Lee? Lee never would have said that to me. Never, because it was, I don't get it. I don't get it. How did you say that I'm looking at this the wrong way around? So I extended this challenge to his 3-year challenge for his assembly theory. I said, I predict that assembly theory will not afford a demonstrative, prebiotically relevant method to make a simple cell that bears the characteristics of life, or generate the specific information that improves that cell, or affords the desired regio and stereoselective glucose dimerization, or selective synthesis of a 200-mer RNA from nucleotides, or selectively afford aspartic acid lysine decay without side chain coupling. I bet it doesn't do it. So these must be predicted by assembly theory and then demonstrated in a laboratory under prebiotically relevant conditions for your assembly to be worth anything. That's what I told them. So if assembly theory can deliver on any one of the 5 challenges within a simple cell, that is the characteristics of life, which are noted at the bottom, I will stop quoting an Origin of Life researcher when he said Origin of Life research is a scam. I will remove all content of my Origin of Life from my YouTube channel and never publicly speak about Origin of Life again. And if assembly theory cannot deliver on any of the 5 challenges in 3 years, and I first put this out on March 4th, 2024 in a YouTube video, then I'll claim that assembly theory is not just a scam, but a grand scam. If he tells me this is going to do something useful and it can't even do these sorts of things, it's a grand scam. Go ahead, prove me wrong. Lee, prove me wrong. I'd love for that to happen. So are we past the hype? Well, no, it keeps coming. Here's an article just recently. We may finally know how the first cells on Earth formed. That's a bunch of nonsense. And I'm seeing some hope here. Here's an article by Nick Lane, an Origin of Life researcher, and his former student, Joanna Xavier, who's also an Origin of Life researcher. And here's some of the quotes. The origin of life is one of the greatest challenges in science. There is no consensus about what to look for or where. Most scientists agree that these nanomachines are a product of selection, but selection for what, where, and how? Refraining from hype might seem unrealistic, but could work if researchers implemented this practice in their roles as peer reviewers for papers and grants, as well as authors. Okay, so they're beginning to, even though they're sheepishly saying it, but they're beginning to say the emperor has no clothes. And Dennis Noble, in this article in Nature, in this past year, wrote, "We are at the beginning of a profound rethinking of how life works. It's time to stop pretending that, give or take a few bits and pieces, we know how life works. Instead, we must let our ideas evolve as more discoveries are made in the coming decades. Sitting in uncertainty while working to make those discoveries will be biology's great task for the 21st century." So Dennis Noble, he's beginning to say the emperor has no clothes. So my suggestion to see progression in the field of origin of life research is stop the overambitious projections regarding the state of the field. Don't abandon the basic reactions of chemistry. You can't just talk about this, your dumb assembly theory, and think that that solves the origin of life questions. Concede that we do not yet sufficiently understand chemical reactions to project them toward life. There are enormous scientific mysteries yet to unfold. They might be hundreds of years away. And the clock started March 4th, 2024, on the 3-year challenge to assembly theory. Good luck, Lee Cronin. Has science explained the origin of life? Not even close. Science might someday do it, but that day is far from today. Well, how do we know that? Because every year the complexity of the cell is harder. Because we learn more about it. Not that it evolves, but that we learn that, oh, I got to deal with chiral induced spin selectivity now. I got to deal with it. So the goalposts keep moving. So we might move a nanometer closer to figuring something out, but the goalposts have moved 10 meters away in that time period. So that's what's happening. It keeps moving further away. And that's how you track progress in a field. You say, how much closer am I to the target? That's how you know you're making progress. But we get further from the target every day. That's how we know we're not getting close. So more realistic targets as an origin of life, say you solve the mass transfer problem or develop prebiotic processes that are 100% yield at every step. Try to solve the 1.0 or 2.0 protein folding problem or interactome problem. The making of certain dipeptides, just try to make dipeptides or disaccharides with selectivity. Try that. The formation of RNA without 2', 5' linkages or branching. Try that. A method to remove human intervention at every step. Every step, you have a ton of human intervention. Try just walking in your lab and looking around and see what comes together without humans trying to push it together. The secret to synthesis can be repeated. It can repeatedly survive impure mixtures or solve the separation problem. Show me chemistry where you can have vastly impure mixtures like come on meteorites and make that do something useful. Even in your laboratory, try that. Solve the relay synthesis problem where you have multi-steps and you're going to keep running out of material. Solve the condensation polymerization challenge where delta G, the free energy, is positive. The polymer stability problem, the time problem. You have hours to a few days depending on the polymer type. It's history. It's toast. That 4 hours was when you had RNA with no divalent magnesium around. 2.5% of the crust of the earth is magnesium. You want to try to have no divalent magnesium around, good luck with that. But if you had it with no divalent magnesium around, you got 4 hours on a 600 mer of RNA. The origin of specified information, where'd that code come from? Nobody knows. Try to think about how you would do the final assembly if you had all the parts. You can even take a simpler experiment. Just because a resurrection is easier than a de novo synthesis. Just take a cell that's just died and bring it back to life. Define for me what it is you just lost when you lost that life. Try that. And then just try bringing that back to life. That's got to be an easier target. Everything's kind of in place. Just try that. That'd be a good first target. So my appeal to all of these researchers is, if we are beset with so many barriers, regardless of the model, why not join me in sharing the obvious with the press and lay public? We have no idea how first life originated. It is obvious that life can be made from inanimate materials. It's obvious. We're here. We are evidence of that. So there's nothing profound in that assertion. We already know it. The problem is that any materialistic model that we invoke is far from having an experimentally verifiable hypothesis as to how life might have originated. All proposals fall far short. We cannot even solve the dipeptide or disaccharide problems with our prebiotic models. And without solving that, you can't solve life. Just make a dipeptide, make a disaccharide with selectivity. If you can't do that, we can't solve life. So, in summary, we remain so far from figuring out how to build even the simplest of cells, and the target moves further away from us every year. Contrary to the claims of these researchers, every step is mind-numbingly hard. It is so much more than just making a strand of RNA. Biologists are sadly confused on this point. We cannot even make the RNA in a prebiotically relevant manner, nor will it last more than a few hours. Realize that people are wrong when they suggest that we're close to solving the construction of even the simplest cell that can be conceived, wherein it meets the minimal requirements of life as we know it. Humankind remains clueless about origin of life, but that does not mean that we will forever be clueless. If you are a graduate student considering a degree in origin of life research, try addressing a problem that can have an impact. Or your professor, you and your future students and their future students will all retire and die before we solve this problem. So with that, I'm going to open it up for questions, I think. So stop right there and open it up.
Thank you so much, Dr. Tur. That was